All right. So how is it that we're getting this volume overload of the heart? So how is our heart adapting to endurance exercise? Well, what we see in a physiological hypertrophy situation, so that's a really important distinction, is that it's a physiological hypertrophy, which means that we're getting what's called a volume overload hypertrophy in the heart. It's different than a pressure overload. A pressure overload is like a pathological problem with the heart. In a volume overloaded heart, you see that the heart holds a bigger volume of blood. You see also that the wall size of the heart is slightly bigger. So you've had hypertrophy of the heart and a bigger chamber size. So the result is the bigger chamber size holds more blood. The increase in the thickness of the wall of the heart allows you to pump out the same percentage of the blood that was there before. So if before you had 70 mils of blood in your heart and now you have 100 mils of blood in the heart and you get 90% of that blood out, that's going to mean that in this volume overloaded heart, this hypertrophied, physiologically hypertrophied heart, you're going to get a lot more blood out in each pump. So our cardiac output is going to go up because our stroke volume has gone up. All right. So that's the real core of this is that the exercise is going to increase our cardiac output by increasing stroke volume as a result of a volume overload, which means the chamber volume of the heart is bigger and the thickness of the wall is bigger so that you pump more blood with every beat. All right. So how has this happened? What is necessary for this? You absolutely require AKT for this. And so here's a beautiful study where they took wild type animals and AKT1 knockout or heterozygous animals, and they either let them rest or they let them swim. Swimming was a nice form of exercise for these animals. So basically what you see is the left ventricular volume here relative to the tibial length goes up when you have the wild type animals with swimming. So you let them swim and whoop, there goes, their heart gets bigger. The volume of the heart gets bigger. If you don't have PKB or AKT, either one copy or both copies, you actually see that you completely prevent the physiological hypertrophy in response to exercise. All right, so then the question then becomes, because PKB wasn't involved in skeletal muscle hypertrophy, we had Rick that was doing everything. So really important kinase was doing this. PKB wasn't really necessary. So that means that our heart is transducing the mechanical signal differently than our skeletal muscle. So how is it happening in the heart? And what's different between the physiological hypertrophy that we see with an athlete and the pathological hypertrophy that we see with a heart disease individual? So if we have a normal individual who's got a heart that holds 70 mils, what happens in a pathological hypertrophy, even if it gets a bigger heart, you get a decrease in ejection fraction, and that's causing a decrease in cardiac output. And so whether it's a volume or it's just the chamber size has gotten thicker, what's happening is that the chamber is getting thicker because the muscle isn't functioning as well. And so there's something that prevents it from functioning. So you get a hypertrophy to try and compensate for the fact that the muscle isn't functioning well. The result is that the big difference is in a pathological sense, you get a decrease in ejection fraction. In a physiological hypertrophy, you get a small increase or the maintenance of ejection fraction. But the result is that in one, you get a decrease in stroke volume, and the other, you get a big increase in stroke volume and cardiac output. Okay? So then let's try and figure out how the molecular signals are different between a pathological hypertrophy, physiological hypertrophy, and what happens in our skeletal muscle versus what happens in our cardiac muscle. So in cardiac muscle, we're going to have to talk about completely new proteins that are involved in transducing the mechanical load signal. So this is a paper that was done looking at knockout animals that had lost this protein, the integrin-linked kinase. So integrin-linked kinase, even though it's called a kinase, it doesn't actually phosphorylate anything. It's lost its kinase activity. But it's still essential to proper cardiac function. You can see that when you knock out integrin-linked kinase, and that's in these MCK-CRE all that means is they use a muscle creatine kinase promoter to knock out integrin link kinase only in muscle, specifically in the heart. What happens to the heart? The heart goes up. It doubles in size. And that's because of left ventricular dilation and impaired function. So without integrin link kinase, the heart grows in a pathophysiological hypertrophy. When they look at this, they did some further work and they said, okay, let's see what happens when we take a normal cardiac cell and we knock down mTOR, just knock down mTOR both in complex 1 and complex 2. What happens? AKT phosphorylation goes up at AKT 473, even though you knock down mTOR complex 2. But that's because you've knocked out this inhibition of IRS1, so you get more AKT phosphorylation at baseline at the 308 site, and even though you've knocked down mTOR from mTOR complex 2, you still get more AKT phosphorylation at the mTOR complex 2 site, simply because there's less inhibition of the IRS1 based work, and so all of the AKT is up at the membrane. So if it's all up at the membrane, even if you don't have as much mTOR complex 2, you'll eventually phosphorylate it all because it's all up there. Because IRS1 is much more active when you've knocked out mTOR complex 1. So now if you knock down both mTOR complex mTOR and Richter, which is the complex 2 important protein, what you see is that phosphorylation of AKT goes down. If you knock out mTOR and you knock out integrin link kinase, now you almost eliminate AKT phosphorylation at all. So there's something about AKT in the heart that where its phosphorylation is regulated by integrin link kinase as well as by mTOR complex 2. And the way that we think this is happening is that the MSIN1 complex that keeps mTOR complex 2 at the membrane doesn't necessarily work as well in the cardiac system as keeping mTOR complex 2 at the membrane. So what happens with the integrin link kinase? As you'd expect, integrin link kinase is connected to the integrins, which are connected to the outside of the muscle fiber. And so what happens is now the integrins connected through integrin link kinase is going to pull PKB or AKT up to the membrane. And it doesn't do it by itself. It uses another protein called myosin. And again, just like with integrin link kinase, where without integrin link kinase you get this pathological hypertrophy, if you don't have if you have too much myosin, or if you have a myosin transgenic animal, what happens is your left ventricular end diastolic volume, so that's how much blood is left after the heart's contracted, goes up because the muscle starts to fail. The fractional shortening rate, or the ejection fraction, goes down. So you're losing functionality of the heart when you don't have myosin. So the way that we think this happens is that now instead of MSIN1 being as important here, MSIN1 doesn't necessarily link mTOR complex 2 as well to the membrane. And so what happens is now the integrins through integrin link kinase and this protein myosin are locking mTOR complex 1, or mTOR complex 2, I should say, and protein kinase B together at the membrane. And when that happens, now you get more protein kinase B signaling, you get more hypertrophy. So instead of you using RIC in the heart to phosphorylate TSC2 and move it away, now you're using protein kinase B or AKT. And it's doing that through this integrin link kinase myosin pathway that's going to connect mTOR complex 2 and protein kinase B, get that whole thing together. So the greater the load on those integrins, the more signaling through PKB there is, and the more physiological hypertrophy of the heart you get. So unlike skeletal muscle, where you have this RXR XX kinase that we call RIC that phosphorylates TSC2 and moves it away, in the heart, you've got this connection between the integrins and the integrin link kinase that brings protein kinase B and mTOR complex 2 together those two things allow protein kinase B to be activated and to signal through TSC2 to drive physiological hypertrophy of the heart. So when you don't have integrin-linked kinase, you don't get physiological hypertrophy of the heart, and the heart becomes pathological. Same thing with Malusin. All right, so this mechanical transduction in the heart is different than skeletal muscle, but it still ends up doing the same thing, turning on mTOR in a way that's now going to drive a physiological hypertrophy.